Hello, everybody, and welcome to today's webinar titled Personalized Medicine, the Role of Pharmacogenomics in Optimizing Drug Efficacy and Safety. This webinar is a part of the Precision Medicine Virtual Event Series. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is sponsored by Thermo Fisher Scientific. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. If you have trouble seeing or hearing the presentation, click on the help desk button from the lobby within the event. Now let's get started. I now present today's speaker, Steve Jackson, Associate Director, Product Applications, Thermo Fisher Scientific. For a complete biography on our speaker, please find it on the left of your screen. Steve, you may now begin your presentation.
Thank you, Sydney, and thank you everybody for being here. As she said, today I'm going to be talking to you about how pharmacogenomics can be used as a personalized medicine tool for helping with drug efficacy and safety. For today's talk, I'm going to start by introducing what the problem is. I'll tell you a little bit about how drugs are metabolized. I will introduce the concept of pharmacogenomics. I'll provide a couple of examples of pharmacogenomics that are currently being used. And finally, I'll enumerate a little bit about how pharmacogenomics can help reduce costs in healthcare systems. So we have been enormously successful in generating new drugs throughout the last, 50 or 80 some odd years. But along with these new drugs come an increase in adverse drug effects. This is tracked by the FDA, as shown on the graph on the right-hand side of the slide here, where the adverse drug effects, or ADEs, have been increasing in their reporting since 1990 at least. You can see that from the graph that the blue is serious side effects, the red is death, and the green are not so serious side effects. But in each case, the amount of side effects that have been reported has been increasing. It's gone down a little bit with the increase of pharmacogenomics, but it's still a significant problem. The CDC has also done some studies on the role or the effect of adverse drug effects, and they found that over 1.5 million people go to emergency rooms due to ADEs each year in the United States, and almost 500,000 of those require hospitalization. Older adults appear to be more vulnerable to ADEs, with more than 600,000 or older adults can visit the emergency departments more than 600,000 times each year, more than twice as often as with younger people. Understanding that the drivers of adverse drug effects can help prevention. And finally, some of the drugs that cause ADEs are anticoagulants, diabetes, and antibiotics, commonly used drugs that are prescribed fairly often. Again, some of these adverse drug effects do include deaths, and so not only is it uncomfortable for the patient, but can also be very tragic for both the patient and the families. These drug-related problems, as quoted in this slide here, can also be costly. This is a study done by a Dutch group that employed a model to assess the hospital costs driven by drug-related problems. The cost for drug-related problems can range from €0 to over 4,000 euros per event. Of all the 676 events analyzed in this study, the total costs of adverse drug effects were over 304,000 euros. This project confirmed that medication reviews performed by clinical pharmacists can prevent adverse drug events, and it can be cost-effective. And they note that pharmacists should focus their interventions on patients that have a high risk of adverse drug effects on the drugs that are being prescribed. Now, the question is, how can we identify these people that have adverse drug events, and how can we find them and define what these patients that have a high risk actually are? So to address this, I'm going to briefly outline for you how drugs are metabolized. And keep in mind, this is a very general overview, but typically what happens are these small molecule drugs are taken by an individual. They're input into the gut by enterocytes. They're transported across these cells into the gut, where they enter the portal vein, get transported to the liver. These are acted on by cytochrome P450 and other enzymes to metabolize the drug in ways I'll show you later on in the talk here. The liver then can export that drug into the hepatic artery for general circulation, hopefully finding the target organ. And then ultimately the drug is excreted by the kidneys where it's eliminated from the body. Now, each one of these different steps can be regulated by drug metabolizing enzyme or DME genes as shown here. They can be found for transport, for metabolism, and for excretion. And because these drug metabolizing enzymes are encoded by genes, they're subject to the sorts of modifications that can happen as part of normal DNA metabolism. Now, some of these drug metabolizing enzymes are grouped into three different phases in general. A few examples are described here. So, for example, phase 1 drug metabolizing enzymes are often the cytochrome P450 or CYP450 enzymes. These are often found in the liver, but can be found in other tissues. They're predominantly monooxygenases that can add oxygen to an atom, making it more hydrophilic. And this can then result in activating the drug and facilitating excretion from the body, or converting an inactive prodrug to an active drug that can then have the desired biochemical effect. Phase 2 DMEs are often transferases, and these are enzymes that are responsible for conjugating other functional groups to small molecules, including drugs. These include the UDP gluconyryl transferases, sulfotransferases, GSTs. Like the cytochrome P450 enzymes, these enzymes help increase the hydrophilicity of small molecules and thus increase the availability of drugs. But these might also produce toxic byproducts as a result of their transferase activity. Finally, phase 3 drug metabolizing enzymes are often transporter proteins. These include the SLC22 transporters, which are involved in moving small molecules between tissues and interfacing drug fluid or body fluids. And there's the P-glycoprotein, which is also known as the multiple drug resistance protein. It's an efflux transporter that influences the absorption, distribution, and elimination of toxins and xenobiotics. So these different families of drug metabolizing enzymes play an important role in deciding how drugs are metabolized in the body once they're given. Now, I mentioned that the drug metabolizing enzymes are encoded by DNA molecules, and as a result, they're subject to the types of things that can happen to DNA molecules. For example, they can acquire single nucleotide polymorphisms, which are a replacement of one nucleotide for another in the coding region of a gene. This can reduce or eliminate function of the enzyme and reduce the enzymatic activity of the drug metabolizing enzyme. Another way that SNPs can affect the activity of DMEs is by making mutations in the promoters and enhancers of genes. The replacement of one or more nucleotide in the promoter enhancer might change the expression levels of the protein, and therefore it can act like a loss of function mutation by reducing expression of the gene altogether. Indels, small insertions and deletions, are loss or gain of function of one of a number of loss or gain of one or a number of nucleotides. This can produce translational frame shifts, which most often produce loss of function mutations, again eliminating the activity of that particular DME. And finally, there are copy number variants where the entire gene can be lost or gained in number. This can increase or decrease the functional levels of the protein by increasing the number of copies of a gene that's present. And therefore, copy number variants are another important way in which DMEs can be affecting drug metabolism. So the study of how these mutations affect the metabolism of the drugs is known as pharmacogenomics. This is illustrated on why pharmacogenomic markers are important on this slide. So as we know, individual responses to the same medication can vary. Within a population, there can be some individuals that have serious side effects of the drug. There can be some individuals that have no response, and a subset of these individuals can also have the desired response. And of course, you want to be able to tell who is going to be able to have the serious side effect, the no response, or the desired response. And so pharmacogenomics is a way of looking at the DNA of these individuals to try to elucidate how their allelic makeup of DMEs can affect their metabolism. So ideally, what could happen is that you could take a DNA sample of an individual, and that individual's DME configuration will be determined. In some cases, the DME configuration will indicate that the drug is perfectly safe and effective at a particular dose. In some cases, that the allelic configuration will dictate that person will need to have a higher dose of the drug in order to get the desired effect. The DME allelic configuration might point to an individual that has severe side effects, and so you might refrain from giving that person a particular drug in an effort to try to eliminate some of the side effects that would be detrimental to the patient. And finally, there are some individuals that might have no effect of the drug altogether. And so for that individual, you might want to give them a completely different drug to produce a desired biological outcome. So again, pharmacogenomics is making use of the DNA sequencing technologies that are available to identify the allelic makeup of the drug metabolizing enzymes that are known to have functions on particular drugs. So the next couple slides will talk about some examples of pharmacogenomics as they're being used right now. So first of all, in oncology, pharmacogenomics is important and is illustrated by tamoxifen. The cytochrome P450 enzyme CYP2D6 can take tamoxifen and metabolize it into endoxifen, which is a more potent effector of the tamoxifen effect. The CYP2D6 allelic configuration affects the amount of circulating endoxifen, as shown on the top graphs on the right-hand side of this slide. In general, CYP2D6, and for that matter, any drug metabolizing individuals, are characterized as either poor metabolizers, intermediate metabolizers, normal metabolizers, or ultra metabolizers. And because they're heterozygous, the allelic configuration can be a homozygous or 2 copies of poor metabolizing enzymes up to 2 copies of ultra metabolizing enzymes. And you can see that for poor metabolizers, a given dose of tamoxifen produces low levels of circulating endoxifen, whereas the ultra metabolizers have higher levels of the circulating endoxifen, and therefore would have a higher level of the actual functional molecule. So knowing the CYP2D6 allelic configuration can dictate how much of the tamoxifen you give to an individual, because of course you want to make sure they get a proper functional dose. But the CYP2D6 allelic configuration can also affect the frequency of reported side effects. So that's shown on the bottom part of this graph here, where again the individuals are shown as either poor metabolizers, intermediate metabolizers, normal or ultra metabolizers, shown by the gold bar. In this particular study, the researchers surveyed people on tamoxifen and therefore endoxifen to find out what their report of side effects were. Individuals that had poor metabolizing alleles had lower reports of unpleasant side effects such as pain, nausea, and ultimately a lower frequency of going off the tamoxifen treatment. Those that had ultra-metabolizing alleles of tamoxifen had a much higher reporting of unpleasant side effects, and ultimately a much higher rate of going off the treatments because the side effects are too severe. So knowing the CYP2D6 genotype can help tune the effective dose and also can help alleviate some of the side effects arising from, in this case, tamoxifen treatment. Another example comes from cardiovascular treatments. Clopidogrel and prasugrel are used to prevent strokes by inhibiting platelet aggregation, and by blocking platelet aggregation, you can block clot formation. Both of these drugs are prodrugs that are converted to a functional molecule by drug metabolizing enzymes. CYP2D6 is one of these, but other CYP molecules are responsible for converting the clopidogrel or prasugrel into the functional molecule. So the CYP allelic configurations then can dictate how much of the prodrug is converted into the actual drug. And so, like with tamoxifen, knowing the CYP allelic configuration can help dictate what the functional dose might be. But it's also shown that CYP allelic configuration can have a greater effect on clopidogrel metabolism than prasugrel metabolism. So again, knowing the CYP2D, the CYP allelic configuration of a patient beforehand can help to dictate which of the two drugs you might want to give that individual. And then finally, for psychiatric treatments, in schizophrenia, risperidone is used to treat some of these symptoms. Risperidone is primarily acted on by the CYP2D6 enzymes to make the active metabolite, but other CYP enzymes and other DMEs can contribute to the actual level of converting the risperidone to the active molecule. Some of the adverse drug effects that arise due to risperidone treatment include insomnia, anxiety, headaches, upper respiratory tract infections, Parkinsonism, depression, and akathisia. So, knowing what the cytochrome P450 enzyme configuration is before giving risperidone can help alleviate some of these side effects. And so, the Dutch Pharmacogenomic Working Group, or DPWG, recommends checking the CYP2D6 allelic configuration before administering risperidone to patients, and they recommend that pharmacists and clinicians check the configuration before prescribing risperidone. So those are a couple of examples of how allelic configurations can affect dosing and functional aspects of the drug. But I'll spend the last couple of slides of the talk talking about how pharmacogenomics can help reduce costs. So first of all, pharmacogenomics can help reduce costs because it can result in faster identification of the correct drug and dose needed to give the desired outcome. This can reduce iterative visits to the clinic or hospitals that are needed to check the progress of a treatment. For a lot of treatments these days, the dose is kind of determined empirically, where you give a guess of a dose. The patient takes it for a while. He goes back to the clinic to find out if it's having the desired biological effect and if it's having any side effects. And based on the outcome of those questions, you might adjust the dose, find out again in a couple weeks after that if it works, and et cetera. Knowing the pharmacogenomic allelic configuration ahead of time might help more quickly fine-tune the dose and the right drug that's needed to give the desired outcome. Pharmacogenomics can also help reduce the chance of adverse drug effects that need secondary treatment. This reduces the need for auxiliary medications and extra visits to a clinic or hospital. And as we saw in the first slides there, reducing the need for, say, anti-nausea medications, reducing hospitalization visits, and ultimately reducing deaths can help reduce the impact on the clinical costs for both the patients and for the providers. Finally, pharma companies can help stratify their clinical trial enrollees to better recognize the significance of the results. So, for example, a pharma company might do a preliminary pharmacogenomic screen to identify individuals that might have a poor metabolizer or an ultra metabolizer phenotype. They can then weed out some of the individuals that might not be good candidates for treatment for this particular drug, and therefore might have a better clinical pool to understand what the effects of the drug actually are, because the pharmacogenomics is known, and they can then go to the market or go to the clinic faster with the drug because they have tuned it to the right pharmacogenomic allelic configuration. This study was done in 2017, so it's a couple years old now, but it does kind of continue to show that pharmacogenomic testing can be cost effective. Basically, they looked at a number of different drugs, and they determined whether it was cost effective or cost neutral, or whether it was too expensive to do a pharmacogenomic test before administering the drug. In the top part of the graph here, you can see that for the different drugs, when you include the cost of the pharmacogenomic test, that a majority of the tests, a majority of the drugs, coupled with the pharmacogenomic test, were cost-effective or cost-neutral, as shown here by the green portions of the pie chart. But even if you exclude the pharmacogenomic test, then the actual cost of administering the drug they found to be much lower, as shown on the bottom pie chart here. So the cost, it was much cheaper to do a pharmacogenomic test before administering a drug to alleviate some of these costs that we talked about. Another example comes from a case of the Kentucky Teachers Retirement System. This is a public health retirement system that was set up by the Kentucky Teachers Group. And as part of their cost containment system, they screened 36,000 individuals as being enrolled in this study to try to include pharmacogenomics as a way to reduce adverse drug reactions in the people that were enrolled in this health plan. What they found was that including a pharmacogenomics test had a 28% reduction in risk-related incidents, a 29% reduction in hospitalizations, and a 24% reduction in just slip and falls due to uncoordination because of the wrong dosing of a drug. Importantly, they found this overall reduced their health costs by 15% in total healthcare costs, and they reduced pharmacy spending by 14%. And they found that overall, by including a pharmacogenomic program in their health plan, they were able to make it cheaper for the enrollees in the system. Similarly, the University of Pittsburgh Medical Center is developing a population scale deployment of pharmacogenomics. The idea is that the clinicians would then perform a pharmacogenomic or order a pharmacogenomic test for patients. They would then communicate with a pharmacist, and then the pharmacogenomics test plus the right medication would then go into a permanent electronic health record that could then be referred to by the health system over time. At the time the slide was made, they had a relatively low number of enrollees at first. It was one hospital and looking at one gene. But as this grows, they hope to enroll over 4 million patients with 40 hospitals and look at hundreds of genes to help control the cost and as we talked about, control the cost of adverse drug effects by including a pharmacogenomic screen into their healthcare, state-run healthcare system. Lastly, the Taiwan Precision Medicine Initiative is a national health plan that is initiated by the Taiwan government. They have a ambitious plan to not only look at pharmacogenomics, but also look at predictive genomics, looking at say, risk scores based on genotypes, et cetera. And so what their idea is to across 16 different medical centers, they would hope to enlist over 1 million participants in this. And they would then not only biobank the materials from these 1 million participants, tissue samples, blood, et cetera, but also collect genomic information from these individuals and help identify pharmacogenomics outcomes by these individuals and also risk scores of these individuals. And their impact is that they would help manage populations that are at risk for common diseases and adverse drug reactions, and therefore reducing the cost overall for their whole health system. So there's several resources available for those that want to pursue pharmacogenomics programs. The CPIC, the Clinical Pharmacogenomics Implementation Consortium, is a group that is involved in collecting information on the pharmacogenomic allelic configuration, drug dosing, and drug prescribing information into one place. This is freely available, and a link is given. It's easy to find the link for this. We don't have a link in this slide, I'm afraid. Another example is PharmGKB. The PharmGKB is part of the HHS and is run by the NIH. This is again a collection of allelic configurations and drug outcomes, and is freely searchable and made available for pharmacists, for clinicians as a source for recommending pharmacogenomic tests for particular drugs. The PharmGKB database is used by a number of different national plans: the Dutch Pharmacogenomics Working Group, the French National Network of Pharmacogenomics, and the Canadian Pharmacogenomics Network. All make use of the PharmGKB information for making recommendations for clinicians and pharmacists in their own countries. Again, this is freely available on the internet and is easy to find if you're interested. So I'll just summarize that adverse drug effects can be detrimental and costly. Many genes, their subject to normal variation, can contribute to drug metabolism. Pharmacogenomics is an analysis of genetic variants that affect a person's response to drugs. And pharmacogenomics is useful for optimizing treatments for oncology, cardiology, psychiatry, and a growing number of other conditions. Finally, pharmacogenomics can help reduce medical costs by reducing iterative visits, reducing adverse drug events, and stratifying likely responders to drug development trials, lowering the cost of drug development in the end. So finally, I thank you for your time. If you have questions, please let us know, and we'd be happy to answer them. Thank you very much.
All right, fantastic. Thank you, Steve, for your time today and for your very important research. Before we go, I would like to thank the audience for joining us today. Any questions submitted will be addressed by the speaker via the contact information you provided at the time of registration. As a reminder, this webcast can be viewed on demand for 1 year until May 14th, 2026. LabRoots, will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everybody, goodbye.